Hello, everyone, and welcome to the webinar on predatory mites as a biocontrol agent for the management of insect-transmitted viruses in vegetable production. This is the third webinar in a series of 4 on biological control of vegetables in the Southeast as part of a NIFA OREI funded project. Dr. Srinivasan is a professor in the Department of Entomology at the University of Georgia. He conducts basic and applied research on thrips, whitefly, and aphid-transmitted viruses affecting several crops in Georgia and the Southeast. So welcome back, Babu, and I'm just going to turn off my video here and hand over the screen to you.
So as Alice said, this is a part of the project, OREI project, that mainly focused on using biological control agents, especially predatory mite, and look at its effect on suppressing pests. And I sort of got involved into this project because I study insect-transmitted plant viruses, and they are a major problem in the Southeast, especially on vegetable production. And so that's how I got involved. And the PI at the time was Rebecca Schmidt from Clemson. And she asked me to check to see how this would be important or useful in suppressing viruses that affect mainly tomato and squash production. So that's what I'm going to talk about today. So there were 3 of us involved from the University of Georgia. That's me, and Alice gave all the information that you need to know about me. And there's also some contact details in case you have any questions or something later on. Feel free to email me. And this is the other co-PI in the project, Dr. Bhavesh Dutta. He's an extension plant pathologist, and he's located at the Tifton campus. And I'm located at the Griffin campus. For those of you who do not know where Griffin is, it's about 45 minutes to the southwest of Atlanta. And Tifton's another 2.5 hours farther away to the south. And so that's Bhavesh. And the other person that was involved with this project was Tim Coulong. He's a professor of vegetable production at Horticulture Department. And he's actually located at the Athens campus. When we started this project, he was actually located at the Tifton campus, but he since then moved to Athens. So, but all of us are still in Georgia and involved with vegetable production, and we've been doing this for the last 15 years approximately. So this project started in 2018, and with COVID in between, so it's been a big hassle, or rather a hustle to get all things done. But anyways, I'll be happy to share some of these results that happened in the last 3-4 years. So what am I going to basically talk about in this talk, right? Presentation rather. I'm going to talk about insects, introduce you to the concept of insects. You all know insects as pests, but I'm going to talk about how they are important as vectors of plant viruses. A vector is nothing but that transmits virus, okay, or any other pathogen, but today I'll just restrict it to viruses. And introduce you to some of the important or economically important viruses in tomato and squash. What are some of the management options that we currently have, and how relevant it is to incorporate or integrate biological control as a management tactic in combination with the others. So let's start with the introduction of insects as vectors of plant viruses. And you might have heard about plant viruses affecting so many different crops, no matter where you are in the country, right? But the point that I want you to take away from this entire busy slide is just 2 things. First, if you focus on the pie chart, more than 2/3 of the viruses that are affecting plants are actually transmitted by insects. And that's what's shown on the right. Insects as vectors of plant viruses, and you have mites as vectors of plant viruses, nematodes, and fungi. As you can clearly see, insects are very efficient in transmitting plant viruses, and some of them are much more efficient in transmitting plant viruses than the others. And there are some reasons for those, and I'll try to explain as we go along in this presentation. Again, there's too many numbers here, but what I want you to focus is on this vector group that's right here. Aphids and whiteflies and hoppers, the hemipterans especially, are the number one group that can transmit the more plant viruses, and that can be severely devastating to either yield and food and nutritional security. The second one that we have to deal with in this part of the country is thrips. Okay, so these are the two groups that I'm going to be talking about today. So first, thrips. So for those of you who don't know thrips, they're really teeny insects, have about a millimeter and a half in length, and they have fringed wings, so they're called as fringed wing insects or fringed winged insects. Excuse me. And they're a major problem, especially in the Southeast, on both vegetable and row crops, right? Especially crops like tomato and pepper, row crops such as peanut and cotton. And these insects have only what we call as asymmetrical mouthparts, in which they have only one mandible, and they sort of rasp on the leaf epidermis and then start sucking its contents, okay, by making epidermal punctures. And it's a hemimetabolous insect, and you can see there's larvae, and then it goes into a quiet stage called a pupae before it becomes an adult. Okay? And there's several species of thrips, and what we concentrate here is the tobacco thrips and the western flower thrips, which is also a problem on the west coast of the US. And so besides feeding, what these insects or thrips do is they transmit a very important virus known as tomato spotted wilt virus. And it's called as tomato spotted wilt orthotosporovirus, but the simple term is tomato spotted wilt virus. And as any name of the virus indicates, it basically is named after the symptoms it causes on the plant on which it was first observed. So it caused spotted wilt on tomato plants or leaves, and then that's why it's called as tomato spotted wilt virus. But it has a wide host range extending to over 1,000 different plant species. And you can see what it does to tomato production on here on the right, and then for peppers. And so this is a huge problem. You can lose a big portion of your crop to this virus. Okay. And so I'm not going to go into the details, but the point that I want to stress here is that different viruses are transmitted by different insects with different mechanisms involved, right? So this is a group of viruses what we call as persistent propagative transmission. And what it means is that once a virus is acquired, it stays inside the body of the insect, and it also multiplies within the body of the insect. Very similar to some of the human and animal infecting viruses that are transmitted by mosquitoes and so forth. Okay. There's a lot of complex interactions involved, but the bottom line is you need to know that there are different modes of virus transmission, and this is one of the most complicated ones. And the other important vector that we have in this part of the country is whiteflies. It's more or less a tropical / subtemperate insect, and it's in the order Hemiptera. But again, the life cycles here, don't worry about it, goes through several nymphal instars, and the last instar is sort of the quasi-pupa stage. And over the years, what happened is we have multiple whiteflies are what we call as a cryptic species complex. What that essentially means is that there are differences, biochemical or physiological differences in how they interact with their host plants, but morphologically or when you look at them, you basically cannot distinguish them. Okay. So we had several other cryptic species in the US, but in the last 30 years, they have been replaced by invasive whitefly species or cryptic species, which is what we call as B and Q. What we originally had was A, and that got displaced by B and Q. And especially B, when it starts feeding on plants like squash, it emits a toxin that causes silvering of leaves. Okay, so that's just by feeding. And they also can transmit several different viruses. Right? So here are some examples of those. The viruses are complex. They could either be DNA or RNA viruses. But here's one. It's called as tomato yellow leaf curl virus. And if you don't plant resistant varieties during the fall period here, you would lose your entire crop. And that's what's shown in the picture at the bottom right here. These are 60-day-old tomatoes, and you can see every single one of them is infected. And the other crop that we're really interested in is squash. And we have several viruses, and some some are DNA and some are RNA. And this one is cucurbit leaf crumple virus. And you can see that's why it's named. It causes leaf crumpling, especially at the seedling stage. And then you don't get anything that's marketable or remotely close to it if you leave it uncontrolled. And there are a few other examples. As you can see. And anyways, these are different whitefly groups. And as I said before, so don't worry about this. So why are whiteflies important to transmission? For those of you who know, viruses require living cells for them to multiply, right? This is why we call viruses as obligatory pathogens. And they just need live cells to multiply. And so if an insect chews on the leaf tissue, and if it completely destroys the leaf tissue, then it's not going to be very effective as a virus host, the plant, after that. So, but instead what these insects do is they make a puncture, and they have what's called as these stylets, which are elongated mouth parts, and they travel through these intracellular spaces in the leaf or the plant until they reach the phloem. And then they suck the phloem sap out and then withdraw their stylets and seal them with what we call as gelling saliva. So the plant remains intact, and while it's feeding, it's injecting viruses into these plants, and these plants can get infected with the virus, right? So they cause minimal damage to the plant, which allows the viruses then to multiply on those plants. And that's why these group of insects, what we call as hemipterans, are very effective in terms of transmission. And they transmit viruses in numerous different ways, in at least a couple of different ways. One is called as circulative persistent transmission. And again, what this means is the virus circulates inside the body of the insect, and once it's acquired, it stays until the insect dies. Okay, there are several examples of those. And then there's also some of these other groups of viruses, RNA viruses. Here what we have is tomato chlorosis virus. And this is also transmitted by whiteflies in a slightly different way, and I'll talk about that in a minute. And here are some more examples. It's another RNA virus that's transmitted by whiteflies, and what we believe is a persistent, semi-persistent mode of transmission. So this is another one which happened. We never knew about this virus infecting squash in Georgia until 2018. So this is or 2016 rather. This is what we call as cucurbit yellow stunting disorder virus, and you can see the symptoms that it produces on squash plants. These are all yellow squash plants or zucchini squash plants. And this one is even newer. It's probably in the last 3 years is when we started hearing about cucurbit chlorotic yellow virus. Also, a very similar group of virus compared to the previous one, crying virus. And they're starting to become a bigger problem here. So these viruses that I talked about, the last three, they're transmitted in what's called as semi-persistent viruses. It means that the virus is only retained for a certain amount of time, and not for its entirety, the insect's entire life, right? And it's retained at the foregut. And what that means is that the insect has to consistently feed to acquire this virus and then inoculate it to a plant that's not infected. And it's a very complicated process. But nevertheless, what I wanted to show you or illustrate to you is that there are these different modes of virus transmission, and there's so many complex interactions that happen between the insect and the virus. And we spent a huge amount of time in our lab just studying these things. But bottom line is, we are able to study these things because of the immense amount of interest that they bring in, because these cause serious losses to vegetable growers in this part of the country, as well as in many other countries. Okay, so so sometimes I talk to you about several different viruses, excuse me, several different viruses in squash and several different viruses in tomato, right? Some of them are transmitted by thrips and some of them are whiteflies. But this sort of cartoon here gives you an idea of the complexity that we're contending with from day to day, right? You can see this one insect can transmit all these different viruses to at least 3 different hosts here, and they sort of become endemic to our production area, which means that they have alternate hosts, especially weeds, which sort of act as a feedback mechanism to the crops, and then the viruses still remain in the system and go back and forth between the crops and the weeds. Okay, so they become endemic here, and we would have to deal with almost all of these every time we produce tomatoes or squash, especially in the fall season. So here's another example. What happens with when you have more than one virus infecting a plant? And yes, you can imagine they become much more susceptible, as like having to deal with both flu and COVID at the same time. We're not going to do that great, right? The same holds true for squash and tomato plants as well. You can see what's here is a plant that's not infected, and what you have here is a plant that's infected with cucurbit yellow stunting disorder virus, and what you have here is cucurbit leaf crumple virus. And what's on D is a squash plant that's infected with both cucurbit yellow stunting disorder virus, CYSDV, and cucurbit leaf crumple virus. As you can clearly see, these plants are not going to yield very well, right? So up until now, what I basically told you or talked to you about is what are the main players in terms of insects that act as vectors of plant viruses. And for this talk, we're only going to focus on thrips and whiteflies, and then how they transmit these viruses and what happens when they transmit these viruses and you have severe losses. Then the most important question becomes, we are part of the land grant mission, and our goal is to reduce the severity of these losses and then sustain our food and nutritional security, right? So what are our options when it comes to managing these serious issues? Well, we could have host resistance, right? Host resistance could be either against the virus or against the insect, or both, if that's available. And different cultural options, which is planting time, using different kinds of mulch, and spacing, and so forth. Chemical options, you guys know that it's mostly trying to do insecticides and sometimes biorationals or biologicals if they're available. And so this is how we've been conducting or producing crops for all these years. And since the viruses became a serious issue, and especially when host resistance was not available, chemical control was the biggest one that people depended on. So in 2017, 2018, when we all got together and were discussing biological control, and they called me and asked, oh, we're planning to use biological control agents, and do you think that will be sufficient to suppress viruses? And my answer at the time was, we're dealing with a very low threshold. For those of you who are familiar with threshold levels, when you're dealing with viruses, there is none. By the time you've seen insects on the plants, it's probably too late, right? Because once an insect is laden with virus, it can probably, once it's past this latent period, can inoculate the virus within a few minutes of feeding, sometimes even seconds. So I was skeptical, but then nevertheless, we are dealing with not just insects, and simultaneously we have to deal with viruses as well, since we're here in this location. So we agreed to look and check to see basically some of these predatory mites would have any effect on suppressing insect populations and subsequently virus transmission. So for the next 10-15 minutes, that's all I'm going to talk about. and I can discuss some of the details as we go along. But host resistance obviously is the number one component, but the problem is we do not have resistance to many of the viruses that we have today. The reason being, some of these are very recent introductions in our cultivated crops, especially tomato and squash, are so far deviant from the wild species that have some sort of resistance. So they are basically sitting ducks when we have some of these virus outbreaks. So again, this slide reiterates some of the things that I just talked about, the challenges with biological control. Biological control can suppress pests and keep them below a threshold level. Threshold level is what basically triggers spraying. If you're a grower, that's what it means. If you go across the threshold, you spray and bring the insects below the threshold level. But if you're dealing with viruses that are transmitted by these insects, there really is no threshold level. So that's the main issue. Excuse me. And then for this particular project, we all used a phytoseed mite, Amblysius swirskii, and we were trying to manage both thrips and whiteflies as well as tomato and squash viruses. Thrips are a major problem for us in the spring season. In squash, on the whiteflies, on the other hand, are a problem in the fall season. So our thrips trials were done in the spring, and squash trials, or our whitefly trials, were done in the fall. And in the fall, we used both tomato and squash, whereas in the spring, it was just tomato. So what we thought, with host resistance available in some cases, we would combine that with biological control. That way we would have more protection. So in this case, it was a thrips targeted trial. We used two varieties, a susceptible one for the virus and a resistant one to the virus. And those are the two names. The Rosa Sicilian is the susceptible one, and the SV cultivar is the resistant one. And we used predatory mite releases, and we had 4 different treatments basically, excluding the control, which received no mites at all. And then the different treatments were mites that were released at the base of the plant, released on the foliage directly, and then we tied the mite sachets to the bottom of the stem as one of the other treatments. And we use the same treatments for both cultivars. Okay. Let's see. And so there are the application levels that we used were basically what was recommended from the company. Cooper, biological control, I believe, Cooper. And we sort of switched between light curative and heavy curative. Whenever I think we mostly resorted to heavy curative treatment, that was the highest. And we try to do it at 2 time points, initially at planting or right after planting, and then 2 to 3 weeks post planting. So what I want to show here, these are thrips numbers basically. For whatever reason, more thrips were found on the resistant cultivar. I'm not sure why. These cultivars were supposed to have resistance to the virus and not against thrips, so we don't really know. But what stood out was that the variety was different, and none of the other interactions were different, and the mites were present in almost all the treatments at equal levels. So mites were present, but for some reason, we had more thrips in one variety than the other. Okay, so when we looked at the virus, right, virus was only observed in the susceptible cultivar, Rosa Sicilian, which was expected because this virus is what we call as governed by a dominant gene, and it produces a hypersensitive response upon infection. So we expected this, and the incidence of the virus was about 15% overall in the years that we did this study. Excuse me, these are the eels that are produced, and once again you can see that the variety was significant, and this is basically what we expected as well. We found more tomatoes, especially what I have here is the large tomatoes that were produced from these trials, and you can clearly see that the resistant cultivar had more tomatoes than the susceptible cultivar right here. Okay, so let's move on, and we did some more trials with whiteflies, and this was done in the fall. And as I said, this was done to target a whitefly-transmitted virus, tomato yellow leaf curl virus. And luckily for this virus as well, we have resistant varieties, but these resistant varieties are slightly different from what I talked about in the previous case. These are governed by what we call as semi-dominant genes, which means they don't offer complete resistance. The plants would get infected systemically and produce symptoms depending on how many whiteflies infest these plants and how much virus is transmitted. And they sometimes heal better under severe infestation and virus pressure, but it all depends on how much virus pressure there is. And how soon these plants get infected. If they get infected really early, then even with using resistant varieties, you might not see much results. So we adapted a similar approach to the last trial. We had the same 4 treatments, applying at the base of the plant on the foliage and using mite sachets. And of course, we had the control. Right? So let's look at some of the results. So what we have here, I believe this was in 2021, I should have labeled it, but these are number of whiteflies that we saw on 5 plants, right? And you can see the number of whiteflies on each of these, on each leaf. It's just sometimes impossible to manage this kind of pressure, right? So what we have here are the whitefly adults in another trial, as well as different treatments, and you also have whitefly nymphs, and you can see there's no difference between whether the variety or cultivar is resistant to whiteflies or to the virus or not. The whitefly numbers pretty much remain the same. These are quite high numbers, and the nymphs were counted on 2 square inch on a leaf. Okay, and pretty much every single plant that we had out there for those years got infected. So at that point, we didn't really bother to collect the yield because we weren't going to get much. And squash is another crop that we are interested in. In fact, the numbers, high numbers that I showed you were actually on squash, not on tomato. Unfortunately for squash, there really is no host resistance. Unlike tomato, either for the TYLCV or TSWV, for squash, there's just absolutely no resistance, especially yellow squash and zucchini squash. So you can speculate as to what would have happened in these trials. Same treatments were applied on the foliage, at the base of the plant, and the sachets. And what you see here are these mites formulated with some sort of green bran, and that's what you see here. And we made sure these mites are alive every time we received them under the microscope before we even apply these things. So again, whitefly numbers, and you can see applying the mites. These are again numbers per 5 leaves. Applying the mites, predatory mites, can reduce the number of whiteflies on a leaf. That's what's shown here. This is over 2 different years. 1 year we found a little bit of significance, and the other year we didn't. But again, you can see the number of adults on a single squash seedling leaf. It's just, if you can count those, I bet there's more than 100 on that leaf. And look at some whitefly nymphs and eggs on each one of those. And keep in mind that this is actually 1 square inch leaf section per fly per week. So it gives you an idea of the tremendous amount of whitefly pressure that we face, right? And you can also see here, there's some reduction in whitefly numbers on some of these treatments when you apply predatory mites. And they do reduce the numbers of these nymphs and adults, nymphs and eggs. But again, I mean, we're talking still about more than 100, 150 or so immatures per 1 square inch of leaf. And all you need is 1 insect to transmit the virus. So it's sort of a losing proposition there. Nevertheless, and you can see the virus infection on each one of these leaves, and every single plant, I would say, were infected and resulting in a complete loss. And this is not unique to the trial that we did. These are problems not just faced by organic growers. Even conventional growers, after spraying 3 times a week, pretty much face the same result. This is just an unfortunate situation that has developed in the last 7 or 8 years in the Southeast, and mainly because of invasive whitefly species coming in, and then the new viruses coming in from other parts of the country as well as from different countries has made the situation sort of unsustainable for especially fall vegetable production. And this is to give you an idea what we're dealing with. We did some sampling based on grids, and what you have here is the amount of whiteflies and what you have at different days after planting. We put sticky cards on some of these plots and monitored the number of whiteflies that landed on each one of these cards. And you can clearly see as time progresses post planting, we end up with this just severe whitefly pressure. And then you have what's below here is viruses present on those grids. And then what you have here is pretty much the same thing to show virus progression over time and time and space. And the darker the color indicates the severity of the virus infection. And you can clearly see almost every single sampling unit has been infected and severely infected, causing tremendous losses. Okay. And this is, like I said, a situation that developed in the last several years, and this trial was done in 2018 and 19, I believe. So what are we doing in terms of managing besides biological control? And there are several things that we've been attempting. One is to use different kinds of mulch, just reflective mulch instead of the standard white mulch that we typically use, and that seems to fare better slightly. And also use live mulch. In this case, we've used buckwheat, and that also seems to help a little bit in reducing the whitefly numbers, at least, but not so much in terms of suppressing the virus as such. The other important control strategy that we've applied over the years is using row covers, and it's useful in some cases and not so, and difficult in the other cases, especially in squash. If you know squash, it's a cross-pollinated crop, and if you cover them with row cover for too long, that's going to affect fruit setting. So we've used row covers at least up until the onset of flowering, and then we remove them, and that seems to help a little bit. And again, the issue then becomes there is no single silver bullet. There is no silver bullet. So how do we integrate some of these things, right? So we've looked at using both silver mulch in combination with row cover. And what we have here is the incidence of cucurbit leaf crumple virus in a grower's field, basically. And what you have is the % incidence. And then when you have row covers, especially, and the amount of virus incidence goes down. And as is compared with no row cover, single row, and we did the same thing with 6 rows, and you can see that. Covered and no covered, you can see the same effect. When it's covered, you have less virus, and no row cover up until flowering, and you have more virus. And if you add the silver mulch on top of that, you can see there's furthermore um, um, minimization of virus incidence as opposed to using the standard white mulch. And you can see close to 70% of the plants are infected if you don't. And the remaining plants basically die. Right. So we've done this over 2-3 years and found similar results. And you still have to continue spraying on top of that. But you can clearly see that protecting the crop, especially at the younger seedling stage, usually about first 2 to 3 weeks since planting, is very, very critical to minimizing virus infection and virus-induced losses. So I think this is probably my last 1 or 2 slides. Basically, I think this probably is my last slide. So what you can clearly see that there are differences in terms of what control management tactics we use, either using a row cover, that's row cover up until flowering. And we use hoops to stretch the row covers out. And then we use live mulch in some cases and reflective mulch in some cases. And as you can clearly see, there are differences, visible differences that explain what we see in terms of management. If you use row covers, it's definitely going to be better than using reflective mulch and live mulch. And the best option would be to combine reflective mulch and row cover. And going back to what was the critical component of this OREI grant was that whether to check using biological control is going to be effective. And what you've seen over the few slides over the squash and tomato trials that we did, biological control has some effect in suppressing pests, right? It could reduce thrips populations, it could reduce whitefly populations. Then the question becomes, is that reduction going to be anywhere sufficient to suppress virus transmission and virus-induced losses? For that, the answer is, well, at least as a silver bullet, biological control is not going to be effective. And what we showed is that biological control can be effective with host resistance, right? If host resistance is available. And that again is very critical. We don't have host resistance for most of the viruses that infect our vegetable crops. So in such scenario, what becomes important is that we're going to have to integrate whatever we have and basically throw the kitchen sink. Biological control is not expensive, so it wouldn't hurt so much to combine it with either cultural or both cultural and chemical tactics. And we use row covers, we use sort of an insecticide planting, maybe if either an organic insecticide or a conventional insecticide at planting, cover them with row covers and biological control, and then continue that after you remove the row covers, right? Or if you're using conventional strategies, you have insecticides that would go along with cultural tactics as well, not so much with biological control. So the bottom line that we're recommending to growers at this point is that you'd have to integrate your management options, and up until at least you have more host resistance. And for those of you who may ask a question, well, why is there host resistance not available for everything? And it's most of the available resistance or genes that confer resistance are in wild species. And the breeder's job is to sort of introgress them or move them from the wild species into conventional crops. And that often is not easy, especially in squash. We're finding that it's not easy at all. And it's going to take, several years before we can even introgress something that's useful and then squash is edible after that. right? So that's where we're at this point. But in the meantime, what we can do is we have a few other options that we can exploit to our advantage. And if we are growing these crops, especially when there's where there is not so much pressure that we deal with, some of these options would be much more effective for you as opposed to us in the southeast. So I'll stop right there and just want to acknowledge my lab group and the funding sources, especially USDA, NIFA, including OREI and SERI for some of this work, and also Southern SARE for sponsoring some of this work. So yeah, I'll just stop there, and I'll be happy to take questions or if there's any concerns.
Okay, thank you very much, Babu. If anybody has any questions about this, please type them into the Q&A box. Can you tell us again, because I know you mentioned this in your last webinar, why you have so much pressure of whiteflies in your particular area? Because I know we had a webinar last week with Oscar Liebert at the University of Florida, and they seem to have somewhat better biological control results.
Okay, yeah, that's a very good question, Alice. So part of the problem is self-made, I believe, because we do grow cotton. Cotton's one of our important cash crops, and we grow over 1 million acres of them. And our squash maybe about 9,000 acres. So what happens in the fall season is that cotton leaves begin senescing, and sometimes, and they also defoliate cotton. And the whiteflies then would basically go and settle on anything that is green. And they love squash and vegetable crops. And they just switch to whatever is available at that point. And unfortunately, it's tomatoes and squash. And it's just the sheer number that emigrates from cotton, then lands on these other crops is one of the reasons, is probably the main reason why we have such high, levels of infestation.
Yeah, and what would be some good ways for growers to be able to predict what management strategies might be most effective in their respective area?
Okay, that's again a very good question, and we're trying to address that. And what we're sort of have come up at this stage, at least for tomatoes, and we're in the process of doing that for squash, is to have a risk mitigation index. And what we do there is if you have a prior history of virus incidents in your field, and it depends on what variety of crop you're planting, we start assigning risk points. And if you grow in a squash with no host resistance in the fall, you start with higher risk points. But then what we also do is provide a whole bunch of mitigatory options, starting with row covers and reflective mulch. And what we're able to do that then is mitigate some of those risk points. And so that's one of the strategies we think might help, at least as sort of a bridge, until we have some host resistance.
Okay, we have a question here. For the TSWV trial, did you conduct it in a field with a history of the virus? They were just wondering if this had an effect on the variety being significant, and when would be the best time in the season, to put up sticky traps for the thrips.
Okay, so these trials were conducted in the university farm, but they were specifically marked for organic production. So there wasn't a whole lot of history in terms of growing tomatoes there before and having spotted wilt, but the other plots and conventional production areas were not too far away from this plot, right? So it's a possibility that proximity had something to do, but producing tomatoes in the southeast and having 15-20% of spotted wilt incidence is what we often take for granted. That's what we're going to deal with, right? And the second part of the question
Yeah,
was,
that was when would be the best time in the season to put up
okay,
sticky traps for the thrips?
okay. If you're wanting to sample thrips, that's assuming that's why you want to put sticky traps. Um, that would be sometime in early spring, say towards the end of April, or mid-April to first week of May. That would be the best time to sample for thrips. Of course, depends on the species of thrips, but most thrips that we deal with here, the tobacco thrips, the western flower thrips, the eastern flower thrips, are pretty much their peak capture dates would fall somewhere between late April to early May.
Have all of these new viruses and invasive whiteflies had a dramatic effect on the tomato and squash production in your area?
Oh yes, it's in 2017 was one of the worst years, and it was the beginning of this, epidemic, and we lost entire fields of squash production, entire fields of squash. They were so bad that some of the growers didn't even want to go spend money picking squash. They just basically plowed them back into the field. It was that bad.
Okay, let's see. I'm just looking to see if there's any more questions. I don't see any right now. I'm just going to give another minute or so, but I just wanted to let everybody know that this presentation is being recorded, and we're having one more presentation next week on biological control from some research out of North Carolina State University. And thank you very much, Babu, for giving this very interesting and informative presentation. And thanks to everyone for joining us today. And if you'd like to learn more about this biocontrol project, you can find more at eOrganic.info/biocontrol.